In this tutorial, you will learn how to annotate a fungi on the Kobo form. In this case, we have two fungi species. We're going to annotate the first one. I select my fungi, and I'd like to know which is the taxonomy or the lineage of my fungi. So to do this, I will go to NCBI Taxonomy. I'll write NCBI Taxonomy, and I select the first one, the first option, and then I select browser. Then you will paste your species right here. You will see that some information will come up, and right here we see the lineage, which in fact is a eukaryote. A fungi belongs to eukaryotes, and we see that it belongs to Ascomycota phylum, and right here we see some other lower phylogenetic levels, such as the family and the genus. We also have some external information resources, and this is important when annotating. These might be useful for finding information to answer your questions on Kobo. So now that we know what is the lineage of our species, we will go to Kobo and start answering our Kobo form. So we paste the species name just as it is. We don't want to change any characters because then it wouldn't match on the Kobo database, and it will look like we didn't submit our species, the species that was assigned to us. So we'll specify the domain, and we already know it's eukarya, and the type of eukaryotes, it's fungi. So here is asking us what type of fungi this is, and is it a lichen? So just out of curiosity, I'd like to see if there are pictures available for my fungi. Pictures appear right here, and just by looking at this, you'll see that, yeah, this belongs to my species, and you'll see that this is a macrofungi just by looking at it. And you know that because you don't need a microscope to look at the fungi. You can see it with your eyes, with your naked eyes, and this is why we know it's a macrofungi. But we need a reference that we can actually put on the Kobo to base our responses. So let's look at the first link that appears right here, and you'll see that this is a database for lichens. So we know that this species belongs to a lichen, and here we have a series of information about the lichen and the thallus, which is actually a specific structure that belongs to lichens. Only lichens have this structure called thallus. And here is telling us some information about the thallus, about the surface, about some other structures, and we are going to use this as our reference for lichens and for macrofungi. So we'll go to Sothero, and we'll paste the link of our database. We'll paste, and we'll copy this as it is right here. We'll go to Kobo and we'll paste it on the reference text box. We'll answer what type of fungi this is. We know that it is a macrofungi, and then we'll answer what type of lichen it is, and is it a lichen? And yes. Here we don't have any comments about this. We'll just put NA, and our full name we'll just put here, test fungi 1. and we submit. And that's it. Here it's telling us our annotation has been successfully submitted. All right, so now we move forward into the next species. And we're going to do the same as we did with the first. We're going to look at the lineage or the taxonomy. We're going to go to NCBI. We're going to paste our species right here. And we're going to see that, in fact, this is an eukaryote that belongs to Ascomycota as well. The family, the genus, and so on. So again, we're going to start by putting on Kobo this scientific name. It belongs to eukaryote. It belongs to a fungi. So now we will answer what type of fungi this is, macro or microfungi. We'll close this, and we will do the same that we did with our previous species, and we'll look at any reference that comes with this species. And we see here some pictures. Let's see what this is. We don't really know. It doesn't seem to be so obvious as the first species. Maybe it could be a micro, but we'll need to use a reference for this. So let's see the first reference here. We don't want to use Wikipedia. Maybe you can use it just to look at it and just for your own curiosity, but we don't rely on Wikipedia. So what we'll do is we will go to the other reference, which is this, which is USDA, United States Department of Agriculture database, and it's talking about pest alert eastern white pine needle damage. So maybe this is giving us an insight that our species is a pest, or it's pathogenic to something, to a tree, to a pine. So let's read about the description of this. So the description is telling us that this species affects white pine needle, it's called, and it's telling us the symptoms, and they consist on yellow and brown discoloration. And now we have an idea that this might be pathogenic for trees, for a host that belongs to a plant. All right, so we'll keep this open and we'll go back to Google, and we'll go to the third reference. So this database called Microbank is giving us a summary of the species. Here also it's giving us the classification. Here we have a description, so we can use this just to find information about, more specific information about the morphology of this fungi. All right, this article, you can see here, it might be difficult to find, but let's see. Here they have a description of this. Let's see if we can also find it on Google Scholar. So let's copy this title and we're gonna search on Google Scholar. Nothing appears. So probably the reference doesn't appear because this is a pretty old description. We're gonna use this, we're gonna read to see what it says. So here it's giving us a description of the species. It's telling us apothecia embedded in browned needles or browned portions. So it's also telling us it has the spores, ascospores. So you need to keep in mind that the type of morphology, meaning macro or micro fungi. It's not explicit on the article. It's not gonna say explicitly that this is a macro or this is a micro fungi. You need to keep that in mind. So we need to understand if our fungi is a macro or a micro based on the literature. Here we have some clues right here telling us this forms ascospores, and ascospores are the spores that members of the phylum Ascomycota form. So if you find these ascospores, it means that it belongs to Ascomycota phylum. If it belongs to Basidiomycota, the spores are going to be called Basidiospores. And here we have a description of the disease that this fungi causes. So needles is meaning the leaves of the tree. So since it's a pine, it doesn't have leaves, it has needles. Okay, we're gonna keep this reference here and we're gonna keep searching. So what we're gonna do is we're gonna go to the useful links right here on the Coda doc, and we're gonna see which references appear for eukaryotes. So we have the first one, which is a Genomes Online database. Let's see what this tells us about our species. We're gonna copy again our species name and we're gonna paste it on the database. We're gonna put the organism and we're gonna put the genus and the species. All right. We're gonna submit, and we don't have any information about our species right here. We're gonna go to the next one, which is GBeef, and we are going to paste our species right here. We have some information, and we actually have a description right here. So here we have the creator or the author of this is the New York Botanical Garden, and that's it. We don't have anything else. Let's see the other one. For this fungi, they found it in the host, which is the Pinus strobus needles. We kind of knew that, but this is a good source of information. We're gonna we're gonna leave that open, and we're gonna go to the next one, and we're gonna go to so this databases tell us about extremophility. This is not something we need right now. Let's see this mushroom expert. All right. So this is actually a description of the genus, of the genera of the fungi. So let's see if we can find our genera. It doesn't seem like it is here. Nope, it's not here. So what we're gonna do, we're gonna go again to Google, and we're gonna see the pictures, and we're gonna find the reference from those pictures. Let's see what this takes us. So we have this website called invasive.org, and descriptor, a sexual spore, and that's it. Let's look at the next one. So we have another one. We have another spore. So we have another picture. We have the USDA Forest Service try to see if they have our species. Here they have a picture of the needles of the pine, and you can see the spots which belong to the fungi. We're gonna go to this one, and here we have disease symptoms and reproductive structures. Our species is the first one, so the publication is right here. You could use this link, this name, and you can find it on Google Scholar. And here we have it. Now we can see the full article, and this is actually a thesis. All right, so we're gonna find our species here. Figure 5, okay. Let's see if we can find an introduction or a description of this species. Further studies show that 3 more fungal species were also frequently isolated from diseased needles. And here is our species. Now we are more than sure that this is a pathogenic fungi that colonizes a plant. Let's see where else they talk about our species. So here we have the references that this thesis reference on the study, and maybe we can find this on Google as well. So we have here a description of our fungi, and we're gonna use the previous reference. It's right here. We're gonna go to Sothero, and we're gonna copy this, and we're gonna select microfungi. We are going to put our first reference right here. We know that it's not a lichen, and it doesn't, in any reference, it doesn't state that it's extremophile. In fact, it says it colonizes the needles of the pines. So this is a pretty standard environment, and it doesn't belong to any consider extremophile biome. So we'll select no, and we know that it's spore forming. So we're going to also citate this reference where it says it has ascospores. We're gonna copy this, and we're gonna go to Sothero again. We're gonna copy our second reference, and we're gonna paste it right here. And we know that it has a microbiome, and the microbiome is the host that this fungi colonizes, and we know it's a pine, and we're going to select plant. If you're actually curious about what this species is, which is the host species, you can look at pictures and see what this looks like. And you can see right here it's a plant, so we select plant on the host. Is it a pathogen? Yes, we know it's a pathogen, and again the host is the plant, and we don't know if it has antimicrobial resistance, and we can actually make a quick search to see in Google Scholar. We are gonna copy again our species just to make sure we don't make any errors in typing, and we're gonna copy, and we're gonna write antifungal resistance. Here we have another, we already saw this, we have another reference, and here is a description of the different diseases that these trees have because this genera of microfungi. So we don't need this reference. We're gonna go to the next one, and it's talking about another species, not our species. So we'll go to the next one. Okay, a community of unknown endophytic fungi in western white. So we're gonna read very briefly the abstract just to make sure what this is about. After we have read this, we're gonna look at what does it mention our species. And right here, for example, the only parasites of P. monticola are Bifusella linearis, which is our species. So here is saying that the only parasite of this pine is our species and some others. So now we are going to search only by the last part of the species name, which is linearis. And this is because sometimes in the articles they don't name the whole scientific name over and over. What they do is they put, for example, in this case, our species is Bifusella linearis, so they will put then B period linearis. So that's why I would like to see if there is more information. So here we have B. linearis. It's talking about our species, on the materials and methods, how they isolated and sequenced this. So here actually they collected the needles of the pine in the United States, and we're gonna go to the next one. Here we have large group of endophytes. So this means that a parasite is inside a leaves. So this is actually another article that we can use as a reference for the for our Kobo annotations, and this is because it's telling us in more detail that this species is a parasite. So we're gonna copy this link. We're gonna go to Zotero again, and an error occurred. So this happens when you paste the link of the PDF and not the link of the article on the journal webpage. What it means is if you paste this link instead of the PDF link, which is this, it will work. Let's have a look. And that's it. We copied this into our Kobo form, and we don't have any information about the antimicrobial resistance or antifungal resistance of these fungi, so we're going to select unknown, and that's it. We don't have any comments, and this is going to be the test 2 for fungi. We're going to submit, and that's it. Our microbe has been successfully submitted. So I hope this tutorial has helped you to understand a little bit more into how to annotate a fungi. There is not much information about some fungi, so it's okay if you have to select unknown for many questions in your Kobo, and just make sure that you have looked into the different useful links or the different articles, and just make sure also that you understand all the definitions for microfungi and what is a lichen, what is a spore, what does it mean pathogen, and so on. So thank you very much.